Welcome to SSE, School of Sciences and Engineering at AUC The American University in Cairo. I'm Rofida Zagloul, a graduating biology senior, minor in chemistry, and pursuing a pre-medical track. And today, I'm taking you through a journey connecting the human brain with the ocean.
Seems there's no link between them, right? Let's see. This research investigates the neuroprotective effect of marine animals' extract, specifically the sea urchins, to treat human neurotoxicity induced by aluminium accumulation within the human brain.
In the toxicological field, human exposure to heavy metals has proven to cause major consequences since the late decades of 19th century. And among these heavy metals, aluminum Al13 is proven to induce human toxicity with high specificity targeting the brain and such acting as neurotoxic agent, which is our focus. Actually, the exposure to aluminum comes from different sources. It occurs through various ones such as air, food, and water. So although aluminum is considered to be a low redox mineral, physiologically it still can reduce oxidative stress or oxidative damage. This can happen actually through different mechanisms, but generally Al can bind to negatively charged brain phospholipids which contain polyunsaturated fatty acids, making it easy to be attacked by ROS. ROS are reactive oxygen species such as oxygen, hydrogen peroxide, and hydroxyl groups. Briefly, neurons are highly sensitive to oxidative stress. Despite the low systemic absorption of aluminum by humans, Al is still proposed to be extensively involved in neurodegenerative disorders and diseases such as Alzheimer's disease and Parkinson's. In this regard, the contribution of aluminum to neurodegenerative disorders was further emphasized upon studying the resulting degenerated neurons. This revealed the aluminum accumulation within different brain parts, especially the hippocampus region with the highest accumulation rate, and thus acting and hindering learning abilities and causing memory loss. So now let me brief you on the literature review considering aluminum toxicity. Investigation of Al toxicity in vitro neurocell lines has been reported in various studies emphasizing aluminum correlation to neurocell death. This, for instance, was emphasized and investigated in 2012 when the antioxidant enzyme's activity in respect to Al showed a decrease in the endogenous glutathione GSH along with other antioxidant enzymes. Additionally, in 2014, aluminum was shown to have the ability to cause not only endoplasmic reticulum stress, but also to produce reactive oxygen species, ROS. The nervous system, on the other hand, is extremely sensitive to oxidative stress as many neurotransmitters such as dopamine and noradrenaline react with the resulting molecular oxygen O2 to generate ROS themselves.
So, these implications appear to be associated with not only the neurodegenerative diseases that are common, but also with the overall cell death and disruption. Actually, this should do nothing but highlight the necessity for further identification of the treatment, which is again the mission of this research.
And here I recall a quote that a professor of mine once told me. Sometimes we have to think out of our feet, so we stepped out of land to the ocean, where the biodiverse marine environment exists.
The ocean is a rich source of structurally unique natural organisms which have recently gained much attention for their considerable biodiversity along with their potentiality in identifying novel drugs. Let me take you back in time to almost 500 million years when the common marine invertebrate sea urchin has existed. Briefly, sea urchin, in Latin known as Diadema Savignyi, belongs to class Echinoidea, subphylum Elytrodoa, and the phylum Echinodermata. What we generally might know is that sea urchins are important herbivores on coral reefs, and in some ecosystems they play a critical role in maintaining the balance between coral and algae. But are they of any benefit to humans? The answer is potentially yes. Sea urchins have proven to exhibit abundant pharmacological effects, as reported in 2014 by Chang et al. that due to the presence of potentially identified biological compounds derived from sea urchin tissues. Such compounds include the high levels of polyphenols that have antioxidant activity. Not only this, but in 2017, Savignyi et al. Obtained the purple sea urchin from the Persian Gulf, which tissues were examined to reveal that these extracts could be used in pharmaceutical industries. So this research is basically focusing on sea urchins and the marine animal of choice, and specifically targeting their shells to be extracted and processed to closely investigate their protective effect in vitro against aluminum-induced neurotoxicity. Sea urchin samples were collected from Ain Sokhna area of the Red Sea in Egypt, weighing 31 to 45 grams in each. The samples were transported alive to the lab where the internal organs were removed, shells were dried at 40 degrees Celsius, then crushed. The crushed shells weighed 304 grams and turned into fine powder that was suspended in pure ethanol 1:1 solution, then stirred continuously in the dark for 24 hours using a magnetic stirrer. The extract was centrifuged at 8,000 RPM for 15 minutes at 4 degrees Celsius. The rotavap device or the rotary evaporator was then used to concentrate the resulting supernatants under a vacuum at 50 degrees Celsius. Now, and since we have obtained the extract, it's time to determine and analyze its potential components. In this regard, GC-mass gas chromatography mass spectrometry is used as it provides an analytical method that combines the features of gas chromatography and mass spectrometry altogether. So we used it to have the extract being analyzed where different substances were identified as shown in this chromatograph, where on the x-axis the retention time taken for the analyze to pass through the column is plotted, while on the y-axis the compound's relative abundance is plotted. The peaks that are shown in here in this chromatograph correspond to the time at which each of these components reached the detector. By further analysis to this chromatograph and the GC-mass report that generated a yield of 31 potentially identified chemical compounds were detected, as shown in this table. As you can see from this GC-mass table, linalool, for example, is a detected compound with the molecular formula C10H18O. It was detected at two different retention times, 4.24 and 8.97, with concentrations 5.67% and 2.62%. According to MPC, Natural Food and Communities, linalool exhibits powerful bioactive properties due to its effect on the serotonin receptor. It helps treat conditions such as anxiety, depression, and also can help to combat insomnia. Several in vivo studies have confirmed various effects of linalool on the central nervous system. Another compound that is present with a high concentration of almost 15.1% is eugenol, with the chemical formula C10H12O2. Eugenol is proven to have a strong potential to modulate neural excitability. Moving forward, the effect of the extract as a whole on cell viability had to be investigated. This was done using the MTT assay. It's a colorimetric technique used as an indicator of cell viability, proliferation, and cytotoxicity. And its detection method is based on the reduction of a yellow tetrazoline salt to purple formazan crystals by metabolically active cells. Our cell line of interest is the SH neuroblastoma cell line. And once the cells had grown and their confluency was clearly observed under the microscope, they were mechanically detached and plated into a 96 well plate. 4 different concentrations of the shell extract were plated in the same wells. Using the microplate reader, the following results were obtained representing the cell viability in each well. The control group had only the neuroblastoma cell line without the extract, and its cell viability is 100%. We found out that starting from 50 microgram per ml of the extract, there was a clear increase in the cell's viability. The highest concentration to be at 100 microgram per ml as shown in the 4th column, reaching almost 50% higher than the control groups.
Another MTT assay is further used to measure viability, but after inducing aluminum toxicity. As shown in this graph, a 250 microgram per ml of aluminum is plated in all wells except for the control ones. The neuroblastoma SH cell line is only added with 3 concentrations as shown in the last 3 columns, 50, 100, and 200 microgram per ml. The results showed that there was a severe reduction in cell viability in the second column that had only aluminum with no extract treatment. It's estimated around 40% decrease in the cell viability. All the cells that had the extract treatment either maintained the control level of cell viability as in the third column or leveled it up higher than the aluminum group with almost 20% in the last one. Last but not least, since oxidative stress is implicated as one of the major underlying mechanisms in a variety of human diseases, it had to be tackled within this research. So 2 different oxidative stress were tackled in this regard, which are NO nitric oxide assay and GSH glutathione assay. In both assays of nitric oxide and GSH, the nitric oxide level was decreased in the shell of 100 microgram per ml, and the GSH was decreased as well in the shell extract as at 100 microgram per ml. And this in fact shows that the shell extract has a protective effect against oxidative stress that is caused by the cells within aluminum-induced toxicity. So this is initially a proof of concept that sea urchin shell extract could have a protective effect on aluminum-induced toxicity in neural cell lines.